Hello science fans and welcome to SHE-ensya. Our topic for today, life tables. Science also deals with the circle of life. In ecology, we are often interested in when organisms are born, up to how old these organisms will survive, when these organisms will die, and why these organisms survive and die at different stages in their life. An ecologist's record of the life and death of organisms is called life history. Apart from being driven by curiosity, life histories are collected so that we can predict the population growth and shrinkage of different species. This is particularly important for organisms that are protected or endangered, while at the same time, in the human perspective, helps us predict how fast our population is growing and how we should intervene to make sure that we do not go much beyond the carrying capacity of the planet. A life table is an ecologist's tool to record the life and death of the species he or she is studying. It helps us predict if populations will grow or decline over time. It factors in different characteristics that would allow a population to grow, such as reproductive rate, the ability of females to bear young, and also the lifespan or longevity of different individuals. There are two types of life tables, cohort and static. A cohort life table focuses on a group of individuals of the same species that were born during the same time period. It documents the survival and reproduction of all members of the cohort from birth to death. In order to simplify the process, cohorts are usually defined annually. So you can start by studying all organisms that were born, let's say, in 2020, and then just focus on them until all members of that population dies. A cohort life table is made by recording how many individuals were born during that year and monitoring how many of them will survive in subsequent years until, again, there are no more survivors in that cohort. This monitoring of their survival is called the survivorship schedule. A static life table, on the other hand, does not track a particular batch or cohort. It looks at the cross-section of a population of different age groups at a shorter time period and records how many of them are surviving per age group. It accounts for the difficulty of tracking all members of a cohort throughout their lives. So, as an alternative, static life tables count how many individuals of each age are alive at a given time. Of course, this relies on the assumption that age-specific survivorship and fertility rates remain constant since the oldest members of the population were born. This assumption can make static life tables a little bit inaccurate because through time, survival and reproductive rates are rarely constant. The use of life tables are often associated with survivorship, and survivorship is an essential aspect of ecology because it measures how well an organism is able to stay alive at different points in its life cycle. Life tables can be used to create survivorship curves. Survivorship curves are graphs showing the number or proportion of individuals of a particular species surviving at each age. It can be constructed for a given cohort or through a group of individuals of different ages based on the life table. There are three general types of survivorship curves, and these often reflect how the species ensures survival. For type 1 organisms, as you can see from the graph, they tend not to die when they are young or middle-aged. They die when they become elderly. Humans actually fall under this type. They usually have small numbers of offspring, and they dedicate a lot of resources to provide lots of parental care to ensure that these offspring survive. On the other hand, you have type 2 organisms that have a constant probability of dying at any age. Survival for them is dependent on acquiring needed nutrition or avoiding predation. Birds often fall under type 2. Finally, type 3 organisms are those that experience very high mortality when they are young. Their curve flattens out as individuals who reach maturity tend to survive for a long time. These organisms produce many offspring but provide little care for them, and that is the primary reason why mortality is very, very high at very early developmental stages. The study on life and death features a lot of math. So at this point, I will be defining three major parameters that feature greatly when we are trying to predict the survival and life expectancy of different species. The first parameter is called standardized survivorship, or L. It is the probability that an individual survives from birth to the beginning of age X. So it is computed by dividing Sx over S0. Sx is a measure of the likelihood the organism will survive at that particular age, which is determined by counting the number of organisms of that species that are alive at that particular age. This is divided by S0, which is the total number of organisms that are born at a particular year. So, usually, standardized survivorship begins at a measure of 1 because it starts with S0 divided by S0. So, for example, if 100 organisms of the same species were born at a particular year, you start out with 100 divided by 100. This goes down over time because, let's say, after 5 years, only 50 of the organisms survive. It turns into 50 divided by 100. And then let's say after 10 years, all of the organisms are dead. It would become 0 over 100. The next parameter we'll be discussing is age-specific survivorship, or G. It is the probability that an individual who has already survived to a particular age will survive to the next year. It is computed by the formula Sx plus 1 divided by Sx, which is the number of organisms surviving in the year x plus 1 divided by those who survived in the previous year. If you look at the age-specific survivorship graphs, you can see that type 1 organisms usually have high age-specific survivorships early on in their life, and then this gradually goes down until the end of life. For type 2 organisms, it is fairly constant throughout their life and then goes down when the species is at its dying age. Finally, for type 3 organisms, it starts out very low, it increases over time and stays flat until all organisms of those species pass away. The third parameter we'll be discussing is called life expectancy, or E. Life expectancy is how much longer an individual of a given age can be expected to live beyond its present age. It has 3 steps for its computation, which begins by computing capital Lx. It has 3 steps in its computation, which begins with capital Lx. Now, capital Lx is the midpoint of survival of each time interval. It is computed by averaging the survivorship of 2 years and dividing it by 2. Step 2 involves the summation or the adding of all midpoint survival values of the species from birth till death. And finally, step 3, which will give us the actual life expectancy value, is for us to divide the summation of all midpoint survival values by the actual survivorship at that age. So as you can see, life expectancy is actually age-specific. It is the expected number of time intervals remaining to members of a given age. Now, when you see statistical values of life expectancy, this is often life expectancy at birth. If you look at the life expectancy graphs of the 3 different types of organisms, you will see different patterns as well. For type 1 organisms, life expectancy also starts very high upon birth, and then again it gradually goes down, very similar to age-specific survivorship. For type 2 organisms, actually starts low and remains constant until end of life. Finally, for type 3 organisms, it starts out low but goes up very high until it reaches a maximum point and then gradually goes down until end of life. In humans, life expectancy has changed over time. During the Paleolithic age, our life expectancy was for us to only reach up to 33 years old. But in a recent poll in 2017, life expectancy for humans now is expected to reach 72.2 years old. However, a lot of factors can affect human life expectancy. Some of the factors that allowed human life expectancy to increase include reduced infant mortality. A lot of children that are being born now are protected and are born in a safe environment. Unlike in ancient nomadic times, children are the most fragile organisms and they can actually die very young due to lack of resources and safety. Other factors also include the availability of food and other needs. With the development of agriculture and also technology, we are now capable of growing more food and providing more food for different individuals. However, because of societal differences, the distribution of food and other resources are not necessarily equal. You also have the quality of health care. When it comes to health care, the better the health care, the longer the life expectancy. And that's why we're now able to reach 72.2 years old on average. But you will also see other societies that have life expectancies that are longer and shorter. Now, if you look at current events, we can see that infectious diseases can also affect life expectancy. The presence of epidemics or pandemics that are new and are difficult to control can also greatly affect our life expectancies. As mentioned earlier, survivorship can also help predict population growth or decline. What you see here behind me are different graphs of predicted population growth of different nations that enable different kinds of birth control. This is going to be a topic in our next discussion on life tables. Thank you for watching this video. And if you have any questions or comments, please don't hesitate to ask me, your resident Filipino scientist, in the comments section below. We will be continuing our discussion on life tables next week, so I hope I can see you then. Please like and subscribe to our channel also. Thank you very much, and see you around!